In this presentation, I want to tell you how we try to innovate and improve our genome diagnostics by introducing long-read sequencing. So why do we want to innovate our genome diagnostics workflow? In our genome diagnostics, we need to detect different types of variants. And for this reason, numerous methods are needed. This is time-consuming, expensive, and sometimes difficult to maintain in knowledge. And the techniques that we use are still coming with limitations. For that reason, we want to replace the techniques array, karyotyping, and FISH with optical genome mapping, and the several PCR techniques that we use beside short-read sequencing for long-read sequencing. So why do we need the PCR techniques besides short-read sequencing? That is because some regions of the genome are not accessible by short-read sequencing alone, and short-read sequencing can be done by mapping. That means that the short fragments cannot be uniquely mapped to the genome. Further, with short-read sequencing there is limited phasing information and direct methylation information is not available. The regions that are difficult to sequence with short-read sequencing are all over the genome and several diseases are involved. So for that reason, we want to introduce adaptive sampling, long-read sequencing into our diagnostics. For this, we designed a panel of genes carrying repeats, genes associated with neurological and epileptical diseases, genes with pseudogenes, and genes affected by methylation, for example Fragile X FMR1 gene. In total, we selected 471 genes, and this is about 1.5% of the genome. Well, how does adaptive sampling work? If a region is of interest, then the full region is sequenced. If a region is not of interest, then the region is at a certain moment ejected from the pore and another fragment can be sequenced. So with adaptive sampling, you have a whole genome sample preparation and a region selection on the pore. So of course, before the long-read sequencing, we need to do a sample prep. We use the short-read elimination kit of Circulomics to get rid of the short fragments before shearing. Then we sheared with the Covaris G-tube. We sheared to a mean length of about 19 kb, and after sequencing this was about 10 kb. This has to do with the preference of the short sequences in the sequence procedure. We know this for sure because one time we ran out of storage and afterwards the mean length was much higher. We did a ligation sample prep with an input of 4 microgram. With an optimized sample prep, we had about 18 GB output, and with that output, 455 of our genes in our panel had a coverage higher than 30x for more than 95% of the genome. And 418 of the genes had a coverage 30x for more than 99% of the gene. And in the previous situation, I also meant of the gene. We started, we started a pilot study for short tandem repeats because we need a lot of PCR techniques to measure this tandem repeats because it cannot be done with short-read sequencing. So why can it not be done with short-read sequencing? That is because the expansion is most of the times not fully included in the short sequence. With long-read sequencing, we expect that the expansion is fully included in the sequence and that we can measure the expansion accurately. We tested 15 samples in this pilot study. It are samples with pathogenic repeat expansions for SCA and Fragile X. And we tried to confirm these expansions with nanopore sequencing. And beside that, we checked if we could distinguish a pathogenic from a non-pathogenic allele with nanopore sequencing. We used our in-house pipeline for variant calling and annotation. This pipeline can be used for short-read and long-read sequencing. Several input files can be used, and we can use this pipeline for SNV, SV, and short tandem repeat calling. For short tandem repeat calling, we use Straggler with a publicly available catalog. Our variant pipeline can be used for pre-processing, annotation, inheritance matching, and there is also a decision tree included. Then we checked with the Straggler catalog. We see the genes on the y-axis and the samples on the x-axis. If we have a variant called for each gene sample combination, and we see that for most gene sample combination this is the case, but there are some issues to be solved, and this is work in progress. Then a look to our results of this pilot study. So in most of the situations we see that the measured repeat expansion is within 2 repeats of the current methods. A couple of times you see it in black, the difference is between 2 and 4 repeats. 2 times you see it in red, there was no variant measured in the Straggler catalog, and about sample 13 we are not sure, because it should be the same sample as sample 3, but the expansions are different, and the expansions are also different compared with the current methods. So we need to do array concurrence check to be sure of sample 13. And we reach the same results with 25% of the reach and 50% of the reach. For SCA, methylation is not really important, but for Fragile X, methylation gives additional information to predict pathogenicity. Well, how does this work? In Fragile X you can have a pre-mutation of 55 to 200 repeats. Then this is an unmethylated situation where the gene can be transcribed. In the pre-mutation there is a risk to pass on to a full mutation in 1 generation, and there is a risk for Fragile X-induced ataxia. However, if you have a full mutation, then the promoter and gene are sometimes methylated, then there cannot be formed a protein that is necessary for brain development and maintenance. In this situation, males are affected, and 50% of the females are affected with Fragile X. If we use Dorado for modified base calling and Modkit for post-processing, then we can see the results in the IGV browser, the methylation results, and then in the control situation you see an unmethylated pattern, you see that in blue, but with the Fragile X patient, with a Fragile X sample, you see a methylated and an unmethylated pattern, and you see that the methylated pattern, which is depicted in red, is most times there in the longer sequences. So now our future perspectives. Our ultimate aim is that we no longer need whole genome sequencing and WES short read for genes in the panel, but for this we need to have a higher coverage to attain to the Dutch diagnostic standards, and because for this reason we need to make a transition towards a Promethean platform, which is installed in our lab now. But with the Minion data we saw already that there is a potential to detect variants that cannot be detected with whole exome sequencing. Finally, I want to thank my colleagues, and I want to thank you for listening. And if you have questions, please send me an email, because I would be really happy to answer them.